Micro and Macroeconomics, Part 3, using the Hardy or studying the Hardy-Weinberg equilibrium. One of the most difficult things to show with evolution is to provide evidences where you can actually see it happening. This is very difficult to see in many different species, primarily because you have to see so many generations come about before there really is any change whatsoever. So in the past, what scientists have done is they've tried to use mathematics as a way of proving, or at least providing evidence to support various theorems in biology. And that's what the Hardy-Weinberg equation does for us. It helps us to identify if evolution is occurring, and in some respects it can even be used to try to pinpoint exactly where and how things are changing, helping us to understand even possibly why things are changing. The Hardy-Weinberg equation focuses on allele frequencies, and we know that evolution is change over time, but what actually is changing is the frequencies of the various alleles within that population. So we know that two alleles, an individual will have two alleles for one trait, and that the expression of those alleles is what determines what the phenotype is. What Hardy-Weinberg, and it was actually identified or created by two separate individuals, two scientists named Hardy and Weinberg, and they both came up with very similar sorts of equations at the same time, or statistical models at the same time, and they were combined together to into one equation that we use today. What Hardy-Weinberg does is it assumes that allele frequencies in a population will always stay the same. So they are in what we would call equilibrium. Therefore, if they're not staying the same, that means that that population is undergoing evolution because the allele frequencies are changing over time. The way that the equation is applied is to look at the allele frequencies, assume that they are not changing. So you create almost what's called a null hypothesis. You assume that this population, the frequencies will not change over time. And when they actually do change, or are from generation to generation, that goes against your null hypothesis, or it negates your hypothesis. And therefore, it's support that evolution is occurring. So it's kind of a backhanded way of giving support for evolution. So the foundations of Hardy-Weinberg are as follows. First off, you need to have one gene, and for that gene, two alleles. Now, we usually focus on a pure Mendelian inheritance pattern, the most basic being dominant recessive inheritance pattern. And in a dominant recessive inheritance pattern, you have two alleles. The expression of one allele will always mask the expression of another. Now, over successive generations, the frequency of each of these alleles, the dominant one and the recessive one, should remain constant. And if they do, this is identified as being in equilibrium. This is what we refer to as the Hardy-Weinberg equilibrium. A population that's in equilibrium will always have constant or the same allele frequencies unless it's being acted upon by an outside influence. So that outside influence being evolution. So all of the assumptions here that things that might change frequency, Hardy-Weinberg has to assume that they are not happening. So these are all the things that we had talked about in micro and macro evolution that might change or might encourage or be part of the evolution of a species. So in order for the Hardy-Weinberg population to be in equilibrium, there can be no recurring mutations, no preferential mating. Everything has to be all random. No differential mortality or fertility, for that matter. Okay, so there's no differential reproductive success at all in that population. There's no traits that provide longevity or ones that lead to early mortality. There's no immigration or emigration, so there's no gene flow at all happening in that population. There's no gene fluctuations due to genetic drift. Okay, so there's no founder's effect. And again, if there's no mutation, then there really shouldn't be any drift. And bottom line is there's no natural selection here. So the environment is constant or remains relatively constant and doesn't influence the lifespan or the reproductive success of any individuals in that population. So for the population to be in the Hardy-Weinberg equilibrium, none of these things can be affecting that population. So that's why we use it, or we term it, the null hypothesis, because we know that this probably isn't occurring. We know that mutations are constantly happening, and therefore all of the population should be changing. But in order to show that happened, we assume that they're not. So, looking at the dominant recessive inheritance pattern, the most basic one, you have two alleles and two phenotypes. So you either have the dominant phenotype or you have the recessive phenotype. The dominant allele always masks the expression of the recessive allele. Now, even though you only have two phenotypes, there are actually three possible genotypes. And it's the genotypes, or the allele frequency, that the Hardy-Weinberg assumes is in equilibrium. So we can have the dominant phenotype, and if it's the dominant phenotype, and if we use the normal naming criteria, which would be a large letter for the dominant allele, a small letter for the recessive allele, someone who has a dominant phenotype could have one or two different genotypes. They could have both dominant alleles, or they could have a dominant and then a recessive allele. An individual, though, who has the recessive phenotype can only have one genotype. So that's the one thing that we focus on here, because if we have an individual that has the dominant phenotype, we have really no idea what their genotype is. However, someone with the recessive phenotype, we know their genotype, and that is little r in this situation, or homozygous recessive for that trait. So, if you only have two alleles in a population, then the frequency of each allele, and that's usually expressed as a %, if I add those two together, the number of the dominant allele and the number of the recessive allele in that population, as a % of the population, I add them together, they're going to equal 1 or 100%. So using a Punnett square and replacing the alleles with the frequencies, what you can see, and this also is a way of looking at the chance of two independent things happening at the same time, are the product of each happening independently. So, if I look at each of these and all the possibilities, which is how the Punnett square was put together to show this, what you can have is that in our offspring, 1/4 of the offspring, okay, are going to have the homozygous dominant trait, which, if I look at the frequency, that's going to be p squared. You're going to have one individual that is heterozygous for that trait. Again, dominant phenotype, but heterozygous. Whoops. You're going to have a second one that is heterozygous for that trait. And then last but not least, you have one individual who's homozygous recessive for that trait. So if everyone, all of these are the possibilities of anything that's produced from this union of two heterozygous individuals, then that has to equal 100%, or everyone in the population has to be one of these four things. Okay. Now, since these are the exact same, I can put them together. And if I look at this, then p squared, okay, the frequencies of both dominant traits, plus 2pq, okay, the frequency of the recessive trait times the frequency of the dominant trait. And since I have two possible ways of getting that, that's multiplied by 2, plus the frequency of the recessive trait squared. Those should also equal 100%. So now you have two equations here. You have p plus q equals 1, but you also have p squared plus 2pq plus q squared also equals 1. So how do I actually use this? Well, when you look at a population, what you do is you want to focus in or hone in on the recessive trait. So here's an example. In our population, the frequency of albinism, okay, so these are individuals that are albino, and it's a certain type of albino, is 1 in 20,000. Okay, this means that Q squared, okay, and that's our frequency of individuals with that recessive trait, that recessive phenotype, is going to be 1 in 20,000. So if I take the square root of 1 in 20,000, I would get the frequency of that recessive allele. And in this case, it's 0.007. Now, in order to calculate the frequency of the dominant allele, all you have to do is use the other equation, p plus q equals 1. Therefore, 1 minus 0.007 is going to be equal to the frequency of the dominant allele, which is 0.993. Number of heterozygotes in that population, okay, is going to be 2pq, or 2 times the frequency of the dominant allele times the frequency of the recessive allele, or 1 in 70. Okay, so now you're going to try your own hand at it. So let's say we have 500 caramel grads, or 250 pairs, mated pairs of caramel grads decided to join an expedition to a neighboring star system. Now, this trip was estimated to take more than 1,000 years. So these people were going to be isolated and away from their parent population for more than at least 10 generations. The ship was, of course, self-sustaining, and in terms of food and water, everyone aboard were healthy and capable of reproduction. After 1,000 years, the voyagers returned, and obviously it was just their descendants that returned, because nobody in this group lives more than 100 years. A number of differences between the crew and the population of Earth were noted. Of the population that returned, there were many, many red-headed individuals. A final count showed that of the 800, so there was a little bit of population growth there, that returned, there were 200 redheads. So, was 200 redheads quite a bit? Had evolution actually occurred? Let's take a look. Okay, so before they left, the number of redheads in the parent population, and this is what it currently is in our human population, is about 5%. And being redheaded is a recessive trait, and it's a one particular gene that if you get 2 copies of it, then you're blessed with having red hair. So let's calculate the frequency of this recessive allele in our parent population. So in the parent population, Q squared, okay, is going to be equal to 0.05. 5% of the entire population had the recessive phenotype. So let's look at and try to find the frequency of the allele itself, just one copy of that allele. So in order for you to get that redheadedness, you needed to have both alleles, both recessive alleles. So Q squared is equal to 0.05. If I take the square root of that, I get 0.22. of the dominant allele, P plus Q equals 1. Therefore, the dominant allele is going to be 0.78. So when the space travelers returned, in their population, 2 out of 800, or 25%, had red hair. So Q squared now equals 0.25, or Q equals 0.5. P plus Q equals 1, therefore P equals 0.05. So can we say that evolution has occurred? Yes, we have, because there has been a significant change in the allele population of both alleles, the allele frequency of both alleles. So take a minute to think about this. Which one of these best explains this result? Was it due to mutation, natural selection, differential reproduction, migration or gene flow, or genetic drift? Okay, once you've answered that, move on to the next slide. Okay, just for fun, it was later found that all on board had a great admiration for their favorite Carmel science teacher, and that those who resembled this teacher were favored over others that did not. Does this change your answer? And if it does, how? Bring your stuff to class and we'll discuss.